So again, we're going to focus a lot on bacterial genetics, but it relates to us too. And actually a lot of the genetics we know is from studying microorganisms, right? Because they're much easier to study. And actually, if you, nobody complains about people working with bacteria. It's when you deal with the cute and fuzzies, people get upset, right? But we do all kinds of mad scientist stuff to bacteria. So for just a quick review, and we've been using this terminology, but I'll give you guys some tips and tricks that kind of helps me out in understanding the terminology as it relates to DNA and RNA and protein. Is if you think about DNA, right, this is deoxyribonucleic acid. It's made up of nucleotides, right, phosphate group, sugar, nitrogenous base, and it's that nitrogenous base, those A's, G's, T's, and C's that we were talking about, that is the code, that's the information part of that molecule. RNA, specifically messenger RNA, is also nucleic acid, right? But it's ribonucleic acid, so it's slightly different. The sugar is a more complete sugar. It's not missing any oxygen. Most of the nitrogenous bases are similar to DNA with one exception, right? We don't have thymine, we have uracil instead. This is molecular-wise very similar, right? They're both nucleic acids. So when we go from DNA to RNA, we refer to this as transcription or a transcript, just as you are doing right now, right? I'm talking, you're writing. Probably still in English, right? Anyone not writing in English? Okay. You're sticking to the same language, right? I'm speaking, you're writing. Same thing with DNA, right? We're going from DNA, one form of the language, to the message, a transcript of that language that's going to then be brought to the ribosome. But the ribosome doesn't speak in nucleic acid, does it? It's going to make what? What does a ribosome make? Proteins. And what are proteins made out of? There are 20 of them that naturally occur. Amino acids. Nucleic acid and amino acids are different, right? Completely different molecules. So that's why this is called translation, right? Because we're going from messenger RNA, which is nucleic acid, to a different type of molecule, you could say a different language, when we go to protein, amino acids that make proteins. So if you think it in that manner, it makes sense that we're saying transcript and translation. Right? So if you went home and changed your notes into Spanish or French or whatever, then it would be a translation, right? Which apparently Facebook doesn't do a very good job with. Some of my friends write in other languages, and I click that button, and I'm like, I don't think that's what they say. It's a nice thought, but I don't think it's very accurate. Who knows? The technology will get there soon. It's frightening. All right, does this make better sense to you guys? Okay, now we're going to go one little step deeper. When messenger RNA is read by the ribosome, it's read in a particular way. Remember last time we looked at transfer RNAs and how they had that 3-loop structure to them? In one end, I called the anticodon. It was 3 nitrogenous bases, 3 of those letters, that match up complementary to 3 letters in the messenger RNA called the codon. So each group of 3 nucleotides, right, 3 nitrogenous bases, is referred to as a codon. This is the code that we have cracked, right, for DNA, RNA, and proteins. So if you think of codons, you think of 3 nucleotides, right, and there are 4 different nucleotides, right? You could come up with 64 different codons, 64 different codes, right? 64 different pin numbers. 61 of them are what we call sense codons. They actually code for amino acids. Oops. How many amino acids did I say occur naturally? 20. But we've got 61 codes. We have more codes than we have amino acids, right? So there's some repetition there. Just like you can have multiple security codes to your house, right? So you can give one to the dog sitter and one to the maid. I wish. I want one of those. Right? And have one for yourself and all get into the same house. The same is true here. There are multiple codes for the same amino acids. And so we'll look at that code. 3 of them do not code for amino acids. They're what we call nonsense codons. And they actually tell the ribosome, stop here. Apparently he needs a stop sign. So this is the secret decoder ring for DNA and RNA. So in the DNA, it would say AAA. In the RNA, it says UUU. Oh, I kind of like that. UUU. The ribosome comes along, and the transfer RNA comes along and binds up with that ribosome that's bound to the messenger RNA, and it matches up, and it says AAA. And at the end of it, it's carrying phenylalanine, that amino acid. So for each one of these, we know, right, that if the messenger RNA says UUU, the ribosome, with the aid of the transfer RNA, is going to put phenylalanine in that position. If it says UAA, it's going to stop. If it says AUG, it's going to put the amino acid methionine there. But why is that one green? It's the start codon. So apparently he needs a go sign and a stop sign, but he only has one go. For bacteria, pretty much all their proteins start with the amino acid methionine. Our cells, on the other hand, not so much, because we modify stuff. Right, we're a little more complicated.
Like for bacteria?
For bacteria. Because that's the one that tells it to start. So that's the first one that gets put in all the time, methionine. Am I going to make you guys memorize this table?
No.
That would be pure evil. That you would dump it once you want to put something else in your brain. What we need to know, right, is how to utilize this information, right? So we know a codon is how many nucleotides or nitrogenous bases? 3, right? 3. So each one of these is a codon, and we know what codons code for what amino acids. So if I gave you a code and I gave you this chart, you'd be able to tell me all the amino acids in order to make that protein, right? And I could even be evil and give you DNA and make you turn it into RNA and then into protein. I won't do that either because it's not a genetics class. But we're going to talk about how this information can change and what effect it has on cells. So you have to understand how the system works, right? In order to understand when it doesn't work or when it gets changed. So in this case, you'll notice for leucine, or proline for that matter, just as long as the first letter is C and U, it can be any one of the 4 combinations in the third position, and it's still going to put leucine there. But what if instead of CU, it becomes CCU? Are you going to put the same amino acid? No, that's now proline. So if in that cell, when they're copying the DNA, they put the wrong nucleotide there, Is there a possibility that the ribosome will put the wrong amino acid? Yeah, you could change just one letter here, you're going to get a different amino acid. But in some cases, doesn't matter if you change this one, you're always going to get leucine. As long as the first two say CU, doesn't matter what the third one is. Because there's redundancy in this code, right? Because again, we only have 20 amino acids, but we got 61 codes. So for some of them, and even leucine, look, it can be UUA and UUG or the CUs, and you get leucine. For other of them, it's like they have 2 codes. That's it. AAU or AAC. I can't remember how to say that one. ASN. What ASN stands for. I don't remember all the 3-letter abbreviations. I should know them. The only flashcards I kept from grad school were the amino acids, because I don't have them memorized. But I had to draw them out on flashcards and be able to recognize all the different amino acids and know their names. I haven't looked at them in like 10 years. But I kept those suckers. It took me a long time to draw them. But I don't use it, right? And again, you guys aren't going to use this, but I want you to understand this, right? Because you can look that stuff up literally in the palm of your hand nowadays, right? On a smartphone. But it's being able to utilize the information that's important. Why is this important? Right? Because the code can change, right? And if the code can change, then the product's going to change, right? If DNA changes, the messenger RNA changes, and the protein changes. Could that be good? No? Sometimes no. Sometimes yes. It's like the lottery, y'all. Sometimes somebody wins, right? Most of us lose. I just quit playing. It's a waste of money. Never going to win. The odds are not in my favor. Or anyone else's for that matter. But somebody wins, so we still play. With this, things change, and somebody wins sometimes, right? They actually get better at something. And so therefore they win, right? Just like when you win the lottery, you have more money. That'd be great. If you're a bacteria and all of a sudden your enzyme works faster than everybody next to you, did you win? You won the genetic lottery. Who in Africa won the genetic lottery recently? We talked about it last class. The tribe that's immune to HIV, because again, they are genetically different. The virus can't attach to them. So this happens, change happens because change can be good. But can change be bad? Absolutely, right? Genetic change leads to diseases that we see, right? I'm allergic to like ragweed and a whole bunch of other crappy stuff. Guess who I got that from? My mama. Guess who I gave it to? My son. Right? It's genetic. The reason why my body responds the way it does to those allergens is encoded in my DNA. That's why my body does what it does. So people who are allergic to peanuts, right? Why is, like, it seems like it's taking over our society right now, right? Peanut allergies, especially if you have kids in school, right? Some schools are like insane about it. Don't give up on me. I'm going quick. I plugged you in. It was not fast enough. You did not warn me soon enough. Basically.